Hello, this is our project for the SURE program in conjunction with Dr. McNeish's lab. My name is Lily Dean. I am a sophomore here on the CSUB campus, and I am a biology major.
My name is Emily Pantoja. I'm a junior at CSUB, and I'm also a biology major.
Hello, my name is Carla Segura, and I am a junior at CSUB and am a biology major.
My name is Jonathan Juarez, and I was the graduate student assistant for the McNeish SURE program.
My name is Micaiah Clay. I am a senior here at CSUB, and I'm a human bio major. Our project that we did for the SURE program is about the effects of plastic pollution on microinvertebrate communities in freshwater ponds. Microplastics are 5 millimeter particles such as fiber fragments of foam that can pollute freshwater ecosystems. This then affects the habitats of aquatic organisms, causing, for example, blockages in their digestive tracts. Microinvertebrate and plastic pollution in freshwater habitats have been neglected historically compared to marine life. Therefore, it is vitally important that it is explored. Aquatic microinvertebrates are big biological indicators of this because they can be affected physically, biologically, and chemically with conditions in freshwater habitats. Most of aquatic life in freshwater habitats, such as chironomids or midges, show short and long-term effects of pollution with cumulative impact.
So our objective for this project is to be able to characterize the macroinvertebrate communities that typically colonize natural tree leaves and compare them to how they interact with plastic substrates. Our predictions include that plastic leaf packs will provide a greater macrodiversity with more unique taxa and functional feeding groups when compared to the natural and mixed samples. And then we also predict that the chironomid larva and pupa will have a greater relative abundance in the plastic samples as compared to the natural and mixed samples. The entire experiment took place on the CSUB campus, and the pond that we used was an ephemeral pond where the water is only there for part of the year. It dries up during the summer months. Our methods began with creating the leaf packs. So each leaf pack had 10 grams of material, whether that was natural leaves, plastic, substrates, or a mix of both. So 5 grams of natural and 5 grams of plastic. The packs were then left around the perimeter of the pond and submerged in the water and were left there for 1 year. When they were collected, they were rinsed, dried, and any detritus and macroinvertebrates that were on them were then placed into sample cups. When we were ready, the sample cups were placed under a microscope and the macroinvertebrates were picked out and then identified to the order level and the family level when possible.
So our macroinvertebrate density is a measure of the number of macroinvertebrates per gram of leaf pack material. We found that there was no significant difference when running a one-way ANOVA in the macroinvertebrate density when comparing the different leaf pack materials. Our figure here shows that data, and the error bars represent the standard error of the mean.
We also calculated the relative abundance of different taxonomic groups across different leaf pack types, which consisted of natural tree leaves, plastic textiles, and mixed samples. We conducted a chi-square test and found that they were significantly unique across all leaf pack materials, where Ephydridae and copepods and chironomids were the most abundant across all leaf pack types. We also calculated the relative abundance of functional feeding groups across all the leaf pack treatments. And using a chi-square test, it was determined that they were all significantly unique across leaf pack materials. Collector-gatherers, which are consisting of Stratiomyidae, Ephydridae, and collector filters, which are copepods.
The relative abundance of Chironomidae larvae and pupae was also calculated, and it was determined using chi-square analysis that there was not a unique pattern in the relative abundance of the larvae and pupae across the different leaf pack treatments.
The results did not support our first prediction in that macroinvertebrates would prefer plastic substrates alone with higher densities and more variety in functional feeding groups and taxonomic communities. We actually found that most variety of functional feeding groups and taxonomic groups were found in the mixed leaf samples, which may be due to there being two substrates, the plastic textile providing habitat and the natural tree leaves providing food for the aquatic macroinvertebrates.
Sorry. We found that the chironomids' relative abundance for larvae and pupae were similar across all the leaf pack treatments, which refuted our prediction number 2. And we predicted that this may have been due to larvae deciding to transition into their pupa stage in the initial leaf pack that they colonized. This suggests that the larvae did not prefer the natural leaves over the plastic leaves.
So macroinvertebrate communities may be using plastic substrates for shelter, habitat, and as a food resource. We did find multiple trophic levels in the leaf packs, which makes this a possibility. This can affect the spread of microplastics to other organisms that are eating those macroinvertebrates. A study by Wilson et al. In 2020 found that there was greater diversity in macroinvertebrates on anthropogenic litter compared to natural river substrates, but found no difference in macroinvertebrate density between those two substrates. Future studies could include longer time frames, variety of freshwater habitats like rivers and streams, and different anthropogenic substrates. And you can also compare terrestrial macroinvertebrate to aquatic macroinvertebrates.
We would like to thank Chevron for providing funding for the SURE program. We would also like to thank the U.S. Department of Education for providing additional funding, the CSUB department, or biological department for providing location and resource services. And we would like to finally thank other collaborators of the McNish Lab for helping with data collection.